Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. Let's continue on our lesson, which is about the production of human insulin. This production of human insulin is one of the application of gene cloning. Let's get started. First step in production of human insulin is isolation. mRNA that code for human insulin is isolated from human pancreatic cell. So, this is the human pancreatic cell that contain the insulin gene. Instead of DNA, we isolate the mRNA. This is because mRNA contain only exon, so no need to take out the DNA. Next, since the mRNA is a single strand, so reverse transcriptase will catalyze the formation of first cDNA. cDNA means complementary DNA. So this cDNA is complement to the mRNA. Next, what happened? This mRNA will be degraded by enzyme ribonuclease. So it will leave the first cDNA. Then, single strand of cDNA is synthesized by DNA polymerase, forming double-stranded cDNA. So this is the second cDNA which complement to the first cDNA. So now we have double-stranded cDNA. Then, this cDNA will undergo adapter ligation by adding restriction sequence at both ends of DNA. So, as you can see, so at the both end of this cDNA consists of the restriction sequence. Alright. Plasmid also is isolated from E. coli. So this is the plasmid, and the bacteria involved is E. coli. The next step is cut. cDNA and plasmid are cut using same restriction enzyme. Produce complementary sticky end. So the example in here we use EcoR1. The third step is insertion. During insertion, this cDNA is inserted into plasmid, open plasmid. And what will happen? DNA ligase will catalyze the joining of cDNA and plasmid so that it will form recombinant plasmid. Now we go to the next step, which is transformation and amplification. Recombinant plasmid is transferred by transformation. It is transferred into this bacterial host cell. Now, these recombinant bacteria or transformed bacteria are amplified to make many copies of recombinant DNA. The last step is screening. Screening of recombinant plasmid using blue-white screening. We already learned in previous lesson, the white colonies contain the recombinant DNA. So we need these white colonies. So then these colonies are cultured. Next, insulin gene will be expressed to produce insulin. So this is the insulin. Lastly, human insulin will be extracted and purified from the E. coli. So this is the product of the human insulin. So we already finished for this production of human insulin using cDNA. Okay, thank you.